Okay, class, back to part two of lecture one. So when I left off with part one, I sort of asked you what was the major energy stores in cells. And, sugars can be stored in glycogen, and you can also have energy stored in the form of lipids and fats. But really, when it comes to doing useful work, the ATP is the main energy source, okay? And the majority of ATP is actually made in the mitochondria through a process called oxidative phosphorylation, which we'll cover at the end of the term. Many of you probably have already had it. But in the case with ATP, the majority of the energy is actually sort of stored in these phosphoanhydride bonds. And if you look, the release of the gamma phosphate here actually yields approximately 3 kcals per mole of energy. And other sort of stores of energy in cells is also in the form of GTP and NAD and NADH as well as FAD. Okay, and so this phosphate here it can actually be used in certain chemical reactions to assist in the process of catalysis. Okay, and it forms intermediates. Okay, so the phosphate from ATP can form these intermediates, and it's basically ATP is used throughout the cell. Okay, so you have your phosphoanhydride bond, which is high energy, and that can actually be added to a substrate. And it's very common for the gamma phosphate to be transferred to a substrate as an intermediate. Okay, and so you can sort of see here, you have this reactant B, and you get an activation step where you get ATP converted to ADP, and the phosphate actually gets added to the reactant. And then in a condensation step, you can actually then form two molecules where it comes in and the phosphate is released. Okay, so this is actually sort of a high energy favorable reaction coupled to an energetically unfavorable reaction to allow this to happen. And it all comes from the high energy phosphoester bond, which helps to lower the activation energy. Here's actually a real world example of that where you can go from glutamic acid to glutamine. Okay, these are very similar. This has a carboxylic acid on the end, and glutamine has a free amine over here. Okay, and so what ends up happening is you take glutamic acid, ATP is converted to ADP, and the phosphate gets added, and you have this high energy intermediate. And then ammonia can come in and displace the inorganic phosphate, yielding glutamine. And so there's actually thousands of similar chemical reactions occurring throughout the cell. This is just one example. And the energy from the gamma phosphate can be used to also join molecules. So it's not only phosphate's not only this great molecule in signaling, it's also vital to many biochemical reactions. Okay, and we'll see that in a second as well as we're looking at glycolysis. Okay, so two other very important energy carriers are NAD and NADP, which form NADH and NADPH. Okay, and this is what's considered an oxidation reduction reaction or a redox reaction. And these are vital for basically moving high energy electrons around. Okay, and redox reactions in their most simplest form, if you want to think about them, oxidation is the loss of electrons, whereas reduction is the addition of electrons. So if you can follow the electrons, you can pretty much follow what's being oxidized and what's being reduced. So NADP up here is actually oxidized, and NADPH is reduced. Okay, and so when these are actually used in a reaction, this becomes an oxidizing agent, and this is actually a reducing agent. Okay, so you can oxidize molecule 1 here by taking oxidized NADP and basically stealing the electrons, so this becomes reduced NADPH. And then this NADPH can actually reduce this molecule and break this double bond and add a hydrogen to the end. Okay, so ultimately some of the things you need to know are these oxidation and reduction reactions. These will become very, very important when we get to sort of central metabolism and oxidative phosphorylation, because NADH is actually the major electron donor in the electron transport chain. Here's just the chemical structure of them. The oxidized form of NADP is here, and when it gets reduced and electrons get added, this proton actually comes in and will get added here. Okay, and the big difference between NADP and NAD is this phosphate is missing in NAD and NADH. Okay, so NAD plus becomes reduced NADH, and NADP plus becomes reduced NADPH. Okay, and so this would actually be a reducing agent, and this would be an oxidizing agent. And the molecule here, which is actually reduced, becomes oxidized, and this molecule here, which is oxidized, becomes reduced. So just know your oxidation and reduction reactions. This is one of those things that I think is important, and it oftentimes finds its way onto exams throughout the course. So please just know this, okay? Okay, and here's sort of another real-world example, and we'll just sort of revisit cholesterol in this. And so in the synthesis of cholesterol, one of the intermediates is 7D hydrocholesterol, and what you have is when this gets reduced, NADPH here is the reducing agent, and it becomes oxidized, and then all of a sudden the cholesterol becomes reduced. Okay, and this brings up two terms that we sort of need to know, and I'm going to hold you guys to these, so once again, don't be surprised if this comes up on an exam. Anabolic versus catabolic. Okay, these are basically two metabolic processes. Okay, anabolic is the buildup of molecules. So example, making cholesterol, versus catabolic, which is the breakdown of molecules, breakdown of sugars in central metabolism. And one of the interesting things about anabolic versus catabolic is the amount of anabolic reactions versus catabolic reactions actually change as we age. Okay, so normally younger people have a lot more anabolic processes going on where they're building up molecules, you know, muscle mass and things like that, versus when you get to be sort of a senior citizen, your body starts to break down everything and all of the molecules and sugars. So both of these are happening all the time in all of our bodies, but there's a shift from basically predominantly anabolic processes as you're growing versus catabolic processes as you sort of age. Okay, so here's acetyl coenzyme A. This is a very important cellular intermediate, okay? And the acetyl group here is attached to the coenzyme A molecule. And this is very important in central metabolism. And the bond between the acetyl group and coenzyme is actually a high energy bond. And the rest of this is all largely just a carrier, coenzyme A. And we'll see in a second how important this is in central metabolism, because this is the intermediate that actually gets fed into the TCA cycle. Okay, there's also ATP-driven catalysis where the inorganic phosphate is not used in the reaction per se, as it doesn't become part of the intermediate, but the energy from the bond is actually used in the reaction. Okay, and so in this case, we're going to go from bicarbonate to oxaloacetate over here. Okay, and so the enzyme that does this is pyruvate carboxylase. And so what ends up happening is you get the the carboxyl group added to the biotin in the enzyme, and in the process it uses the energy from the gamma phosphate of ATP and converts the ATP to ADP in inorganic phosphate is released, so that you have this sort of high energy intermediate. And then you can basically add the carboxyl group to pyruvate and you'll be left with oxaloacetate. So in this instance, certain reactions can occur without adding the phosphate to the intermediate, and it just uses the energy in the gamma, the energy in the gamma phosphate to actually help carry out the reaction. Okay, back to condensation and hydrolysis. Okay, so in the case with condensation, you can join two molecules and release a water, whereas hydrolysis, you can break two molecules and water is consumed. Okay, so this is energetically unfavorable and this is energetically favorable. So if you were to just leave a chemical reaction or this substrate A and B out at room temperature, it's eventually going to form this, whereas if you had this just left at room temperature, it's not actually going to form. So the condensation is energetically unfavorable. And so once again, when a water molecule is released and a bond is formed, that's considered condensation, and the addition of water and the actual consumption of the water into a new breaking of a molecule is considered hydrolysis. And so the question then is, when do these things come into play? Right? They're happening in the cell all the time. Condensation reactions and hydrolysis reactions happen all the time. Okay? And some of the most notable condensation reactions are if you're basically generating polysaccharides. Okay, so you can add a glucose molecule to existing glycogen, and you basically get a condensation reaction and water is released. Okay, this happens when proteins are synthesized. So when you add an amino acid to an existing polypeptide, there's a hydrolysis, or there's a condensation reaction where water is actually removed. Okay, and you basically form the peptide bond between the amino terminal end and the carboxy terminal end. And this also happens with nucleic acids when you're adding a nucleotide to either RNA or DNA, and basically you get a condensation reaction where water is released. Okay, hydrolysis is the reverse of this, and in the case with nucleases and proteases that sort of break down RNA or DNA, RNA, and proteins, they will actually consume the water and use the water to actually break the bond in hydrolysis. So ATP can also be hydrolyzed into pyrophosphate. Okay, and this becomes very important in nucleic acid synthesis. Okay, so here we just have ATP, and if you hydrolyze it and add water to it, you can break off a pyrophosphate, and then the pyrophosphate is then further hydrolyzed into inorganic phosphates. And in the end, you're left with adenosine monophosphate. So you can go from ATP, hydrolyze that, to AMP and pyrophosphate, and then from the pyrophosphate down to two inorganic phosphates when the pyrophosphate gets hydrolyzed. And so the question is, when does this happen? Okay, and it actually happens in DNA and RNA synthesis. Okay, so nucleoside monophosphates right here are actually converted into nucleoside triphosphates. And so ATP comes in, and the pyrophosphate can be added to the high energy base, and then the pyrophosphate can get is released when this gets added to a polynucleotide chain, okay, like DNA or RNA. And then eventually the pyrophosphate is hydrolyzed into two inorganic phosphates. Right, and we'll cover this much more when we're covering basically DNA replication and DNA synthesis. Okay, so in the case with condensation reactions, you can have either head polymerization or tail polymerization. Okay, and so head polymerization occurs on proteins and fatty acids, whereas tail polymerization occurs with DNA, RNA, and polysaccharides. And basically it just means where this is being added. Okay, is it being added to the head of the growing chain, where 7 here is added to 6 and then 8 is added to 7, or it's added to the tail. Okay, and so depending on the subunits being added, it can occur on either end of the polymer. Okay, now on to a quick primer with glycolysis. So glycolysis is the conversion of sugars to pyruvate. Okay, so here you have one molecule of glucose, which is then converted into 2 molecules of pyruvate. And this is actually part of central metabolism, okay, where you basically get this one glucose molecule with 6 carbons on it, and it's converted to 2 pyruvate molecules with 3 carbons. And in this whole process, 2 ATP molecules are consumed in some of the early steps, and then in the later steps, you get basically the production of 4 ATP molecules and then reduced NADH. Okay, and these 2 NADHs can then also be used for energy, as can the ATP molecules. Now, in the absence of oxygen, glycolysis goes through a different path. Okay, so if you don't have a ready supply of oxygen, glucose actually gets converted to lactate. Okay, in this process. So here you have glucose, and through the whole glycolysis process, you're left with 2 pyruvates. Now, because there's no oxygen around, this sort of reduced NADH has to be regenerated into the oxidized form, okay, so that it can continue this whole process. And in order to do that, the regeneration process of NAD actually converts the pyruvate into lactate. And this oftentimes happens to runners and other athletes, and can lead to cramping and acidosis or lactic acid buildup. And so that's what's happening when, you know, marathon runners are running and their oxygen supply, you know, they're exerting more energy than there is enough oxygen around to do it. And so the lactate can build up, and you can get lactic acid buildup, okay, and acidosis and cramping. Now, yeast do things a little bit different, and you guys know from last lecture how much I love yeast, and this is actually one of the reasons I love yeast, because it actually makes alcohol when it ferments. Okay, so in glycolysis, if there's no oxygen around, what yeast will do to regenerate its NAD is, once it's converted the glucose into 2 pyruvates, it actually then will create acetaldehyde, which is then converted into ethanol. Okay, and then this is actually how they ferment a whole bunch of different things, from beer and wine, and then, you know, other hard alcohols are further distilled. But ultimately, fermentation in yeast produces this ethanol, and one of the things that I'm sure you guys have all seen is a lot of times you'll see t-shirts where it says Ben Franklin is quoted saying, "Beer is proof God loves us." That's not actually true. The actual quote is down below, where Ben Franklin said, "Behold the rain which descends from the heavens upon our vineyards, where it enters the roots of the vine to be changed into wine, a constant proof that God loves us and loves to see us happy. And a lot of scholars think what he meant by this was, that the rain was proof that God loves us, and not the alcohol, although I'm sure he's pretty pleased about the alcohol as well. And one final note with alcohol and ethanol, as we sort of mentioned, you can have back reactions, right, in pretty much everything. And so when ethanol is being broken down, it's actually converted to acetaldehyde. And acetaldehyde is actually a carcinogen or a genotoxic stress. And so it's widely known now that alcohol consumption and high levels of alcohol consumption can actually contribute to diseases like cancer. And that's so just something that you guys should be aware of. Okay, so let's sort of just focus in on glycolysis. And this is pretty detailed for you guys, and I don't want you to necessarily memorize individual biochemical reactions. These are sort of things that I think you guys should be able to look up anytime you need to know them, but I really want you to understand the sort of the big picture, how things are happening, right? And we've seen different examples of how different chemical reactions happen and how enzymes catalyze those reactions. Okay, so in this instance, when we look at glycolysis, if we want to focus just here on step 6 and 7, and this is the production of reduced NADH, as well as the generation of ATP. Here's how the process happens. Okay, so you have glyceraldehyde 3-phosphate, and then you have your enzyme glyceraldehyde 3-phosphate dehydrogenase, which has a free sulfur group which comes from a cysteine within the enzyme. Okay, and we saw when we were looking at the amino acids, one amino acid cysteine actually has a sulfhydryl group, and so you have this sulfhydryl group. And what ends up happening is you get a covalent attachment between this sulfhydryl and the glyceraldehyde 3-phosphate. Okay, and in the process, it's taking in the oxidized NAD, and then it releases a reduced form of NADH. Okay, and a proton. Okay, and then as step 6 goes on, you have this sort of high energy thioester bond here, which then is easily displaced with inorganic phosphate. And then the phosphate is now attached to the glyceraldehyde 3-phosphate to make 1,3-bis phosphoglycerate. Okay, and then it's this high energy phosphate is then transferred to ADP to generate ATP in this step here, step 7. Okay, so step 6 produces the reduced NADH, and in step 7 you're actually producing ATP by the transfer of this high energy phosphate onto ADP. And in the end, you're left with 3-phosphoglycerate. Okay, and then this will eventually get converted to pyruvate. So if we take a closer look at it, which I think I did a good job on that slide, but here you have the glyceraldehyde 3-phosphate, and with the free sulfhydryl on the enzyme, it actually forms a covalent bond, and in the process, this oxidized NAD gets reduced, and then you have this high energy thioester bond, and a phosphate comes in and can displace that, so you're left with 1,3 phosphoglycerate. Okay, and in this case, you're releasing reduced NADH. And then this high energy phosphate on step 6, which is part of the 1,3-bis phosphoglycerate, it actually gets added to the ADP to form ATP. And then you're left with 3-phosphoglycerate. Okay, so the high energy bond, the phosphate is transferred to the ADP to form ATP. And so in the end, what you end up having is an aldehyde and reduced NADH and ATP, and then you get the formation of a carboxylic acid in the process in those reactions. And so the way this all happens is it actually couples favorable reactions to unfavorable reactions. Okay, so free energy goes up as NAD goes from its oxidized to its reduced form, and in the process the CH bond oxidation energy is driving that, and you get formation of a high energy phosphate bond, and then this high energy phosphate bond is then a downhill reaction to transfer the phosphate to ADP to generate ATP. Okay, so it's the coupling of these reactions that allows the formation of this high energy NADH and the high energy ATP. Okay, so something that sort of should be in the back of your heads too is that the different phosphoester bonds on proteins have different energies. Okay, and so here is basically the anhydride bond in like ATP, and when it's hydrolyzed, it can release, you know, about minus 7.3. And as you can just see, as you go up, certain ones have much higher energy when they're broken. Okay, and so it's just important to know that pyrophosphate, for example, releases more energy than just the release of the gamma phosphate. Okay, so now on to other energy stores. And so in the case with sugars, these can be stored as glycogen, right? And we already saw the condensation reaction that can occur where the glucose can actually be added to glycogen. And these glycogen stores are oftentimes within the cell. This is just a nice scanning image of a liver cell, microscopic image, and here you can see the glycogen granules within the liver cell. When the liver needs energy, it can take that and hydrolyze the glycogen down into glucose, which then can be converted into pyruvate via glycolysis, which we just covered. Energy is also stored in fatty acids, where you can have sort of a fat droplet, and what you end up having in these fat droplets is sort of this triglyceride here, where you have the hydrocarbon tail, and excess sugar intake can be converted into these fatty acids, and then it's stored in fat cells called adipocytes. And both glycogen and fatty acids can be used in the TCA cycle. Okay, and so if we look here, you can take in foodstuffs as sugars and polysaccharides, and then you know, the sugars can be stored as glycogen and eventually then broken down into glucose, and then through glycolysis it's converted into pyruvate, and then the pyruvate can then be converted into acetyl coenzyme A. Same thing with fats. Fats and fatty acids can come in and get stored in fat droplets, and then they can actually be broken down and converted into acetyl coenzyme A. Okay, and the acetyl coenzyme A is then used for the TCA cycle. And here's the TCA cycle, and I'm sure you guys have all seen this at one point or another. Here you have your acetyl coenzyme A, and the two carbons from the acetyl group get added to oxaloacetate to form citrate, and then as everything is sort of happening here, you're basically releasing reduced forms of NADH molecules and GTP, and also reduced FAD, okay, as the cycle goes around. And it's this reduced NADH that's actually used for the majority of the ATP synthesis that occurs in the cell. Just a note on GTP and FAD. So GTP is very similar to ATP, but instead of adenine, you have guanine here, and then you have your GTP with the alpha, beta, and gamma phosphate, just like with ATP. And once again, GTP is actually used in a lot of different reactions, as we'll see. So it's involved in translation on the ribosome, and it's also involved in signaling with small G proteins, and it also regulates various processes like, vesicle formation in the secretory pathway, as well as vesicle fusion. FAD is somewhat similar to NAD, and here it is in its oxidized form, and as you add electrons, it gets reduced, and specifically the electrons get shared across here, and you get the actual protons added to it. Interesting thing about FAD is that in some organisms it actually acts as a redox sensor. Okay, so it can be used to sense oxidative stress and other things like that. And so here's basically glycolysis in the citric acid cycle, sort of from a 10,000-foot view. You get glucose, and the glucose goes to glucose 6-phosphate, down to fructose 6-phosphate, and down finally to pyruvate, and then to acetyl coenzyme A, and then it enters the citric acid cycle. And what's important to know here is that, the TCA glycolysis and the citric acid cycle are really sort of this central component of all of central metabolism, okay, because from this a lot of different cellular things are made, including the nucleotides, amino sugars and glycolipids, and lipids, amino acids and pyrimidines, cholesterol and fatty acids, glutamate and other amino acids, and the purines, heme and chlorophyll, which we'll revisit later, and then other amino acids like serine and alanine, aspartate, as well as some other things. And so in this process, you're really only releasing 2 net ATPs in glycolysis, right? And then the majority of the citric acid cycle is just producing the reduced NADH, okay? And so the question then comes, where does all the ATP come from? Okay, if this is central to producing ATP, where does it all come from if it's mainly producing reduced NADH? And the answer is oxidative phosphorylation. Okay, so if we look at a mitochondria in here, what you have is the pyruvate from glycolysis coming in, and that gets converted to acetyl coenzyme A, which then the acetyl group enters into the citric acid cycle, and every turn of the citric acid cycle, you basically get carbon dioxide released and NADH produced, okay? And it's this reduced form of NADH and the high energy electrons that are then shuttled into the electron transport chain for oxidative phosphorylation. Okay, and we'll cover this later in the term, but these high energy electrons eventually create a proton gradient, and the flow of the protons through ATP synthetase actually converts the ADP into ATP, and in the process these electrons eventually end up on oxygen to convert it into water. Okay, so the reduced NADH produced by the citric acid cycle is used to drive ATP synthesis, and for every molecule of glucose, approximately 30 molecules of ATP is made. Okay, and so in the end, what you have here in central metabolism, and we saw all the little offshoots here where you can get serine and threonine and other amino acids and fatty acids and lipids and all kinds of other things, but here is basically glycolysis, where a sugar comes in, and then eventually it gets converted down to pyruvate and then into acetyl coenzyme A, and then it enters the TCA cycle. Okay, and one of the things I just want to mention here is there's a huge new resurgence in the interest in central metabolism. And this course is not a course on metabolism. There are other courses available for that. But, you know, you can sort of see metabolic syndromes. These are a huge problem in the United States, and there's, a lot of costs to medical care due to the obesity epidemic. And you can sort of see this and the importance of it with the Rutgers Institute for Food, Nutrition, and Health, which is actually one of the centerpieces here on the Cook campus. And so I'm sure you guys, a lot of you have maybe eaten in the cafeteria there, and they're really trying to promote healthy eating and healthy lifestyles, as well as help to understand how this all feeds in. Okay, so that's all I have for today. For next week, just read ahead of time Chapter 3, which is on proteins. Those of you who've taken Dr. Kahn's biochemistry course, 403 and 404, this should all be really straightforward. And review the material I covered in class today, and because we'll revisit central metabolism later in the term. Specifically, we'll go into really great detail on oxidative phosphorylation. And oxidative phosphorylation and the electron transfer chain is one of these areas of science that I really love, so it'll become very important for you guys. And when we do that, we'll actually see in depth how the cell actually converts the reduced NADH into ATP. Okay, so that's all I have for today, and I'll shortly upload these and you guys will have them. So have a great day, everybody. Bye-bye.